Okay. Yep. So there's a reason why I have it that way. We're going to come back to this slide several times. Okay. So the last little bit of this chapter I want to talk about really sets this up for the genetics chapter, which we'll be starting next class. So I want to talk about 3 different aspects of meiosis that lead to genetic diversity. And these are kind of things that I've glazed over. I haven't really mentioned in detail yet. And this is going to lead to why we see the genetic patterns that we see as we go through this. The first one is actually something we're going to kind of ignore for the first half of the next chapter, and we'll only talk about it kind of near the end if we have time for it. And it's something you would consider a little bit more if you take a genetics course, because it complicates things, is something called crossing over. So during prophase 1 of meiosis 1, your homologous chromosomes don't just line up next to each other, they actually can swap a little bit of genetic material. So I'm going to come back to this slide for the other ones. But here's crossing over. So here's a pair of homologous chromosomes, a pink and a blue chromosome. During prophase 1, those homologous chromosomes will line up next to each other, and they could actually swap a little bit of DNA. So now this sister chromatid here has a little bit of the pink chromosome on it, and the blue one has left a little bit of on the sister chromatid that was mostly pink here. The result of this is after meiosis 1 and meiosis 2, instead of having 2 entirely blue chromosomes in those 2 cells, you now get one that's entirely blue, but you get one now that's what's called a recombinant. It has mostly blue chromosomes, but has a little bit of pink chromosome there. And the opposite has happened here. You get a recombinant with a little bit of blue and a little bit of pink. And of course, this doesn't just have to happen once. It can happen multiple times, and you can actually get a double crossover event where you'd end up with a blue A here, a pink B, and a blue C, or something like that. You can end up with many different versions. It could happen on the bottom side here as well. It could happen on opposite sister chromatids. And so it allows you to have many more combinations. So right now, instead of just having 2 different cells produced here, so 2 blue and 2 pink, you now get a blue and pink, a pink and blue, and a pink. So you get more combinations. So how this is going to relate to genetics is, here is going back to homologous chromosomes where I labeled potential kind of genetic traits. So if you got a chromosome that came with freckles, brown eyes, and blood type A from your mother, and you got a chromosome with no freckles, blue eyes, and blood type O, without crossing over at all, you would either pass on this chromosome and therefore pass on freckles, brown eyes, and blood type A to your offspring, or you would pass on this chromosome, no freckles, blue eyes, and blood type O. But because of crossing over, if there was a crossover event here, you could pass on no freckles, blue eyes, and blood type A, or a chromosome that has freckles, brown eyes, and blood type O. So it allows you to have more variety in what you pass on to your offspring. And actually, as we get into genetics, if we have time for this, we'll talk about how you can actually use offspring and potential of offspring having certain traits to actually map a chromosome and tell where genes lie next to each other, because it's actually the ratio of how many individuals have the parental sequence and how many have the recombinant sequence is based on that. So that's something we might get into in the next chapter. It's something you would definitely get into if you took a full-on genetics course. So crossing over in prophase 1 of meiosis 1 can lead to recombinant chromosomes that have portions that have been swapped between the homologous pairs. Okay. So that was number 1, crossing over.
Isn't the yield of both 2 parental and 2 recombinant? Like, don't you yield to, after crossing over, you get
Yeah.
2 parental
you'd get, yeah, so that, the picture I showed before had an entirely blue and an entirely pink. So you had two parental sperm cells, perhaps. And then you had two recombinant sperm cells where one was mostly blue with a little bit of pink and one was mostly pink with a little bit of blue. So you'd have, the recombinants are the ones where the genes were actually swapped, and the parental are the ones where there was no, so the sister chromatids where there was no swapping. But what I was saying is you can get swapping in multiple locations, you can get multiple crossovers, you can get all kinds of varieties of that. Right. We might talk more about that in genetics when we get to that chapter. Okay, so that's number one. Leads to lots of variety, which means that not all sister chromatids are going to be identical anymore. Second thing is independent assortment. So during metaphase 1, your homologous chromosomes will line up on the metaphase plate. So here's an example of how they might line up. Here's a blue versus a green chromosome. So in this case, a blue and a green are on each side. So the blue from this pair's on this bottom side and the green's on top, and here the blue's on top and the green's on bottom. Independent assortment says that homologous pairs line up with random orientation. So it would be just as likely for this to happen as for the opposite to happen, for there to be two blue on one side, and two green on one side and two blue on the other side. So I have a graphic which displays this a little bit better. So that's independent assortment. Independent assortment says that during metaphase 1, your homologous pairs randomly orient on the metaphase plate. So what that allows to happen is this. So here are simple cells that have 3 chromosomes. They have 6 chromosomes, 3 homologous pairs of chromosomes. In this version, all of the blue chromosomes lined up on the top of the metaphase plate and all the red ones on the bottom. In alternative 2, homologous pair 1 and 2 lined up with the blue on top and the red on bottom, but pair 3 lined up with the opposite orientation. In alternative 3, 1 and 3 lined up with the blue on top and the red on bottom, and 2 was in the opposite orientation. And in alternative 4, only 1 had the blue on top and the red on the bottom, and 2 and 3 were in the opposite orientation. And so now you end up with, from alternative 1, this is ignoring crossing over now, so you'd add that extra bit on top of this, you end up with an entirely blue and an entirely red sperm cell, perhaps. If alternative 2 happened instead, you would end up with sperm cell with 2 blue and 1 red, and here one with 2 red and 1 blue. Alternative 3, you'd end up with the same kind of thing, but with a different red and a different blue. And you'd have alternative 4, where you have another combination. So here you have 8 possible outcomes from these 3 homologous pairs. And think about how this magnifies when you have an organism like us with 23 homologous pairs, all lining up with random orientations, which means just independent alone leads to an immense number of possible outcomes. And then you add on top of that crossing over, which means that you can have swapped just little bits of DNA from each of these. We now have a lot of different combinations. So crossing over, independent assortment. And then finally, after you've produced these sperm cells and egg cells with crossing over and independent assortment giving you these random possible combinations of your genes into your own sperm and egg cells, it's random which sperm meets up with which egg cell. So which of the thousands of sperms that are produced each day in a male, which of the possible outcomes of combinations that are produced in the egg actually come together and are fertilized. If you add up all the probabilities, there's somewhere around 70 trillion possible diploid combinations between just 2 people. So this means that 2 parents are very unlikely to have identical kids, ignoring identical siblings, which we'll talk about in a minute. So you're not going to have 100% of the same genes as your brother or sister, because there was a 1 in 70 trillion chance of that happening. Yep.
But that's possible, right?
It's possible. Winning the lottery is possible. Getting struck by lightning 50 times is possible. But pretty unlikely, yeah.
Has it ever happened?
Not that I documented case of this happening. No, two siblings having 100% identical chromosomes. You're going to share some things. You're going to, because the pool you're randomly picking from is the same. So you're going to share a large number of genes. And the possibility of you sharing about 50% is pretty high. But the possibility of sharing 100% is 1 in 70 trillion. Right? So very, very low. So kind of a testament to this are fraternal twins. So fraternal twins are 2 sperm cells, 2 egg cells, fertilized, and you can have a male and a female. So this means one of the sperm had an X and one of the sperm had a Y and produced a male and a female offspring. You can end up with 2 daughters, 2 sons that are fraternal, but they're going to share some similar traits. They're going to be the same age, so maybe that contributes to fraternal twins sometimes looking similar. But as for do they have all the same genes? No, not at all. They're just as likely to have the same genes as any other sibling combination. Right? So fraternal twins are 2 separate events. It's actually the rarity of this happening is because most women don't release 2 egg cells each month. So you have to have 2 ovulation events at the same time, and this is what leads to this. Yeah, so it happens in some families. It's more often because women that tend to ovulate from both ovaries each month tend to ovulate from both ovaries each month, and they have a high chance of having fraternal twins. Right? There you go. So, yeah, all right. So very rare, but So do you have a twin? All right. Are you the same as your twin? 100%? No. So just like any other sibling, right?
He's tall, I'm short.
Yeah, there you go. So there's just one trait, but I'm sure there's many traits you don't share. So it's because it's completely random. It would be very, very unlikely for the same sperm and the same egg. So the only way that you can have 2 individuals with the same chromosomes is if this happened, which is how identical twins are formed, which is 1 sperm, 1 egg, producing 1 zygote, and then early on, first few divisions of that zygote, first few mitotic divisions, the cells don't stick together, and they form 2 separate embryos. And at that point, since none of the cells have differentiated, you can still create 2 entirely functional whole beings, and you end up with 2 embryos that end up with identical twins. So that's because you're not going to get the same sperm, you're not going to get the same egg. Fraternal twins are not going to be the same. Yeah, you could. That would be very, very, very rare. But it could happen that it splits once and then one of those 2 splits again. You could end up with quadruplets that way if they don't split. But it's rare enough for the zygote to split and have separate cells, but it'd be even, yeah. Certain fertility procedures can enhance that likelihood. Oftentimes, product of things like IVF, twins are going to be fraternal because you're implanting multiple embryos. All right. So here's a question just to see if you've been paying attention to these last 2 slides. The phrasing in this is from your book, so it's not me that came up with this pair and a spare thing, which I don't know if that's actually a thing. But so many sets of triplet consist of 2 identical and 1 fraternal, basically. Yeah, I'd never heard that, but this was the slide your book made up. So, so how many sperm and how many egg cells are required to produce this? To have 1 set of identical twins and 1 non-identical. There's 3 total.
Okay.
So triplets, I said many sets of triplets, which means 3 offspring, are not 3 identical. There's 2 that are identical and 1 that is a fraternal twin. 2 that are 2. So, okay, all right, good job. So to make identical twins, how many sperm and how many egg would be involved? 1 sperm and 1 egg, because you have 1 sperm and egg forming 1 zygote. That zygote then splits to be 2 embryos, right? So that was 1 sperm, 1 egg, 1 sperm there, right? There you go. Good drawings. Okay, so how about to create the fraternal twin, the extra one? You would have to have one sperm, one egg, to make one zygote, to make one embryo there, right? So you end up with 2 sperms, 2 eggs. Right? So good, most of you got that. So the only way to have 2 individuals with identical chromosomes, identical genes, is for them to have come as a product of meiosis producing the same sperm and the same egg, because the chances of having that same process occur twice to create the same combination of sperm and the same egg, and to have those 2 actually be the ones that are fertilized, is 1 in 70 trillion. So these 2 will be the same. This one will be different than those 2, right? So you end up with, right. I don't know of any possible way that you could have 1 egg or 3 eggs and 1 sperm, or 2 sperms and 3 eggs, or 3 in 1. It does. In humans, that's not possible. I think there are other organisms that might be able to have things like that, I know, but there's a lot of actual biological processes that prevent multiple sperms from getting into 1 egg or things like that.
Theoretically, what would happen if multiple sperm got into an egg?
You would not have a developing offspring, so it would not survive. Well, this happens. Like the egg will be surrounded by sperm trying to get into it. As soon as one gets into it, it's called blocks to secondary fertilization. Basically, the egg will secrete chemicals that kick all the other sperm away and prevent them from going any further. So once one gets in, that's it. If 2 were to get in and actually fertilize that egg, it would be triploid. It would no longer be diploid, and it would probably not survive the first round of division. So a woman would never know she was pregnant because it would not go any further. So it's a good question, but yeah, it just doesn't happen. At least as far as I'm aware, that would not be survivable. We're going to talk about some survivable weird things that can happen. There are, just like kind of very rare events that are survivable. But most kind of crazy things you can come up with what would happen when this happens, the answer is no one would be born like that. It wouldn't survive. It wouldn't develop to the point of even implantation. The zygote would not survive. Right? Okay. Okay. So difference between mitosis and meiosis. This is something I would like you to know, to be able to compare the 2. Mitosis, the goal was to make an identical cell to your parent cell. So if you started with 4 chromosomes, you'd end up with 2 cells with 4 chromosomes. It goes through prophase, metaphase, anaphase, telophase. You are separating the sister chromatids. Meiosis, on the other hand, the goal is to cut the number of chromosomes in half, to go from a diploid cell that had 4 chromosomes here to 4 haploid cells that all have 2 chromosomes. Meiosis, again, occurs in 2 steps. Meiosis 1, where you're separating homologous chromosomes. Meiosis 2, where you're separating sister chromatids, all of which leads to having 4 cells produced each time.
Can you repeat mitosis?
Mitosis, the goal is to make an identical cell at the end. So you start with 1 cell, you want to end up with 2 cells that are identical to the parent. So goal is to make identical copies. Right, so you're separating sister chromatids in mitosis. So you're not cutting the number of chromosomes down. So the big difference in meiosis is what happens in meiosis 1. Meiosis 1 is what's unique. It separates homologous chromosomes, cuts the number of chromosomes in half. Meiosis 2 is actually very similar to what happens in mitosis. You're separating sister chromatids. Also remember all the things that lead to variation in the chromosomes you end up with, including crossing over, including random alignment during metaphase 1, so independent assortment. That leads to having 4 cells, yes, but all 4 of these are different. So, yeah.
Mm-hmm. Now, if you're making a sperm cell, right,
Mm-hmm.
so you want the chromosomes in half, so you go from 46 to 23.
Mm-hmm.
Now you have that sperm cell that has 23. does that undergo mitosis or meiosis, or
No.
that just stops?
This is it. It will grow a flagella on it, but that's still, it's going to be one cell. Sperm cell is a single-celled
It won't
thing.
undergo mitosis.
Yeah, it does not. In humans, no. There are other organisms where that does happen, where this cell will undergo mitosis to create a multicellular haploid individual. That's what happens in mosses, happens in seaweed, happens in some ferns, things like that, but it doesn't happen in humans. In humans, this stays as a single cell until it reaches an egg, which was also a single cell. They combine, they produce a zygote, right? Yeah. Sperm and egg are both haploid cells. Those are the only haploid cells in humans. Yeah.
They can only combine with each other. They can combine
Yep. With themselves? No. Well, it's a good question, because in some organisms, they could. So two egg cells could combine. There's possible, right? Yeah, and then it would be self-fertilizing. There are, I wouldn't rule that out as a possibility of something that could happen. But has it ever happened in humans? Not that I'm aware of. So does it mean that it would not be possible? I don't. Theoretically, sure. There's a lot of possibilities, right? But yeah, there's a lot of biology that would prevent that from happening. But why not? You've got two sets of genes you could recombine. All right. So meiosis versus mitosis. Okay, so now we're going to talk, the rest of today's class, we're going to talk about some problems during meiosis and what that would lead to in the offspring. So meiosis, when it functions properly, produces 4 cells, all of which have single chromosomes and has half the number as the parent. Sometimes there's mistakes during meiosis. One of the mistakes that can happen is something called non-disjunction. And during non-disjunction, which can happen during meiosis 1, it could happen during meiosis 2. So if it happens during meiosis 1, then homologous chromosomes are not actually separated from each other. If it happens during meiosis 2, then the sister chromatids, they split, but both of them go to the same cell. Non-disjunction means that you're going to create some cells that have too many chromosomes and some cells that have too few. So this is just looking at one homologous pair. And if this homologous pair separates and there's a non-disjunction event during meiosis 2, then instead of these sister chromatids separating and going to one to each cell, like they did up at the top here, both of them go to one cell and none get passed on to this cell. So again, just looking at one pair, so this would be a cell that only has 2 chromosomes in it. If this is the egg, and these eggs all get fertilized by a normal sperm cell, the 2 on the top are normal. They are diploid zygotes. They have 2 copies again, one of each, or one homologous pair. But on the bottom, we now have 2 abnormal zygotes. We have a triploid zygote, has 3 copies of this chromosome, and we have a haploid zygote, has only 1 copy of the chromosome. Typically, in humans, a haploid zygote will, again, not survive. It will probably not go through 1 round of division, definitely won't go through many rounds of division, probably won't implant. The woman wouldn't even know she was pregnant. Right? Oftentimes as well, triploid zygotes do not survive. Most chromosomes, if they are triploid, are not survivable. We're going to talk about some cases of survivable triploid, some chromosomes when they're triploid can be survived, and we're going to talk about that in the next few slides. So this is a problem of during meiosis of the chromosomes not actually separating. It's really a problem of the machinery maybe breaking down, and maybe the spindle didn't completely attach to both sister chromatids, and so both got pulled to one direction. This actually occurs with some regularity. It becomes more and more common the older you get. So the older you are, the more likely there is to be a problem during meiosis and the production of sperm and the production of egg leading to non-disjunction. So older parents tend to have these next few traits we're going to talk about in their offspring more often. Doesn't mean it can't happen in younger parents. It just means that it's more rare, and you think of how many eggs and how many sperm are produced, the chances of them coming together and leading to this, and it being a survivable version of this, are pretty rare for younger parents, but becomes more common as you get older. You have a question?
What is non-dysfunction? You said that's a failure of meiosis 2. Would that just be a failure of, would it be considered like meiosis 2 complete failure, or just prophase 2, because prophase 2 doesn't have it?
Well, it would be a failure. It could be a failure of the spindle fibers attaching, which happens between prophase and metaphase. So it could be a failure of one of the spindle fibers attaching, and so that both sister chromosomes are now pulled to one side rather than being pulled to opposite sides. So yeah, it could just be a small change like that, like a spindle not attaching. Yeah, yeah. So right. Okay, so here's one case where this is survivable. So if it happens to happen on chromosome 21, the offspring can survive. So chromosome 1, chromosome 2, chromosome 3, offspring would not survive. Chromosome 21, offspring can survive, and this is called Down syndrome. And Down syndrome, having an extra copy of chromosome 21 leads to many different problems down the line. So there's developmental problems, there's so a lot of brain development problems, and so it leads to some common traits which you probably are aware of to some extent. So I think the amazing thing is how this is a survivable trisomy. It's really kind of an odd thing. So Down syndrome children tend to be more common with older parents. Doesn't mean they can't be born to younger parents. But what this is a failure called non-disjunction during meiosis. And it could happen with either parent, really. Okay, so that's one that's survivable. Here's some other ones. People always are interested in different kinds of sex chromosome disorders. And here are 4 versions of this. So this would be non-disjunction between the X and the Y in the male making the sperm, or the X and the X in the female. So you could end up with an individual that is XXX, so triplo X. This is actually 1 in every 1,000 to 2,000 females, so pretty common. This is something that you might never know about because it is something that does not cause drastic differences, but slight differences. Women with 3X chromosomes tend to be taller. They tend to have menstrual irregularities. They tend to have normal IQs that might be lower than their family members, but normal IQ ranges. And they are going to be more likely to have egg cells with abnormal numbers of X's, because how do you split 3 in half? You get 2 in one and 1 in the other. So they're going to be more likely to have XXX and XXY offspring because of that. But it's something that you might never know about. How many times have you had a karyotype done of you to look at the number of X and X chromosomes or Y chromosomes? And you probably never, and most people never would, right? Because it doesn't have something that's going to really drastically alter your life. So that's called triplo X. You can also have XXY, which is Klinefelter's or XXY syndrome. The syndrome, this is also much more common than you might have thought, and it varies widely as to the effects of this. Usually there's some underdevelopment in terms of sexual characteristics. You could also some irregularities in body type, but again, something you might not be drastic enough for you to notice it. It can also lead to some learning disabilities, but usually not drastic, right? So again, something that is common in the population and people might not be aware of it. There are XYY individuals, so this could be non-disjunction in the sperm that leads to a sperm with 2 Ys being passed on. And these males would be pretty much apparently normal. They might be very tall, and they tend to have acne problems, but there's lots of other causes for being tall and having acne problems. So again, you could be a male that's XYY and have no idea, and you might never ever know this. And then there's Turner syndrome, which Turner syndrome actually is a lack of a second sex chromosome. It's only one X. And one X women are usually shorter, usually underdeveloped in terms of sexual characteristics, but normal intelligence. They tend to be infertile, but they can take hormone supplements to promote growth and sexual development and can develop normally. So this is they only have one of the two chromosomes, and for some characteristics, some biological processes, women need genes produced from both X chromosomes to have a normal quantity of certain proteins. So having only one X chromosome producing those RNA copies means they're deficient, but they still have those proteins, so they tend to be less developed, which means that if you take hormone supplements, you can be absolutely normal, and even without them, you might not really ever know. So lots of different combinations, and this is all, again, due to non-disjunction. So it seems like non-disjunction in sex chromosomes, non-disjunction in chromosome 21, survivable, non-disjunction in most other chromosomes, not survivable.
Where does it happen most often? Does it happen most often in sex?
Well,
Just one sex? You
I don't know. It's really, the thing is that we don't know how prevalent it is in most of them because they're not survivable. And you think of how many sperm and egg cells are produced that never get fertilized or never go under fertilization. So we have no idea how often they've had that. And then out of the ones that do get fertilized, how many don't exist, don't actually, so we really don't know. This is the same thing with if it takes, having unprotected sex once, you could get pregnant, but chances are pretty slim because there's so many ways that the sperm or the egg could be abnormal, and so many of them that come together would not produce a viable offspring, that the chances of that happening, it makes it kind of baffling how often people get pregnant from one the unprotected sex or one broken condom or something like that. Like, it's really amazing that it happens so often because if you think about all the possible things that could lead to there not being a viable offspring. It's pretty amazing. All right, so that's just some ones that you might be interested in. Yeah.
The XO, does that lead to like PCOS and whatnot?
You know, it could. There's lot for that. There's lots of problems with that. PCOS. That's because that is often a lack of certain hormones being produced enough. So I don't know enough about it to know for sure, but I wouldn't be surprised because that does. Yeah. Yeah. There's lots infertility issues. There's so many different causes due to so many different things that's why it's such a hard thing to treat. It can be really difficult. And if it is this, it's actually something that it's nice to actually have a reason, and you could actually treat that by saying, oh, you just need more of these certain proteins, and you could treat that, right? Okay. So, let's see, where am I? All right, okay, here's a question based on this. All right, Turner syndrome. Turner syndrome is a female. It's that last one, the XO. It's a female that's born with only one X chromosome, so no second X chromosome. Where did the non-disjunction occur? Does it occur in sperm formation or egg formation, or could it occur in either? Yeah, I needed it again. But so let's say, can it happen? Non-disjunction of the sperm. So here's XX, XY. You get normal meiosis to make two eggs. You get non-disjunction here, and you get nothing here. So you end up with X, nothing. So yes, it could happen with the sperm, right? Could it happen with the egg cell? So XX, XY, you get normal XY, you get non-disjunction here, and you get X blank. Yep, so you could happen with non-disjunction by the XX, so the answer would be Either, C, either sperm or egg. So again, non-disjunction from the male could lead to XXY. Non-disjunction of the egg could equal XXX or XXY. So the only one that you could not get through non-disjunction of the sperm alone would be XXX. So you wouldn't get that. But all right, yeah, so really it could be either parent. So it's not just a problem for older mothers, could be a problem for older fathers as well. The reason why we tend to focus on mothers is that males produce so many sperm each month that the chances of that one being is pretty low. There's so many variables that go into that. Females put more investment into the eggs each month, so they're, I don't know. But it is a possibility with either one, right? Okay. So, all right, the last thing really with this chapter, and we'll stop, we won't go into the next chapter today, is just to say that errors occur constantly within chromosomes. They can occur during copying the DNA, during mitosis. They can happen during meiosis with separating the DNA. And they can happen during S phase, which happens prior to meiosis as well. There's lots of different chances for mutations that can have major effects on an organism, or can have major effects on a whole population of organisms. And if you take 1011, you'll actually, hopefully they go into this. I did when I taught 1011, is you can go into how this leads into things like evolution. There can be things. So here's a normal chromosome. You can have gene deletions. So crossing over is not always even. You don't always cross over all the same genes. So you could end up with one chromosome having more and one chromosome having less genes. So if you had crossover between two genes like this, you could end up with one that has A, B, C, D, and you have one that has A, B, C, D, E, F, D, E, F. So you can have a gene duplication in the second one because it gets two copies during crossover event. You can get inversions where the genes get flipped up in the order, and that could lead to differences in expression of those genes. You can get translocation, so genes put from one homologous pair to the other, things like that, so they can end up with differences like that. Things like gene duplications, this actually leads really well into evolution. If you think about, let's say gene D here produced some essential protein. If gene D was deleted, that zygote would probably not develop. It's an essential protein. Or that individual would not survive very long, would definitely not reproduce and have offspring. So a gene deletion or a mutation in D is not going to be very common. But let's say we have a duplication of D. We now can be free to mutate gene D and still have a functional version of it. So you now have one that is able to be mutated and one that's able to be read normally. So this would be like you all of a sudden get two copies of your textbook. You can now write all over one and you still have one perfect copy that you can sell back or something like that. Right? So it's free to mutate. And this actually makes it very easy to come up with a novel protein. So it's very hard to conjure a protein out of nothing through mutation because you need a promoter that works. You need a promoter that binds to certain transcription factors at certain times. It reacts to things appropriately. You need start and stop codons. You need all that stuff to be present. And through random chance, it's very hard for that to happen. But if you have two genes that are functioning normally, one of them just starts changing slightly, changing a few amino acids here, a few amino acids there, you can now have two functional proteins that maybe now have different functions to them. So mistakes, although most of the time lead to non-viable offspring, not viable proteins, lead to apoptosis, things like that, where you're going to get rid of that cell. They can also lead to chances for mutation that might be advantageous. So if you do take 1011, you might talk about this as a mechanism for evolution, and this would lead right into that. All right, so next class, we're going to see how all these possible outcomes then lead to genetic traits. So we're going to start in Chapter 10, Genetics, on Wednesday. All right.